Okay, so today we're going to kind of introduce a topic. The next topic of the class is going to be about data expression regulation. So, first of all, we'll talk about the kind of the conceptual nature of this. So, if we go back and look at the history, in the 50s, it was considered that DNA was a genetic material. You all have read about those experiments from Bio 1. And so that's the person coming down in genetics. From that early work through the discovery of RNA and then the discovery of the ability for RNA to direct protein, we came up with this scheme right here. And so what this says is that DNA produces an RNA, and then the RNA is used to produce proteins. Now, that's been our kind of standing thing for the last half a century or more, probably. Okay? But let's talk about where these terms that we use come up from. So, DNA makes RNA, which is transcription, right? And then RNA makes protein, which is translation. Now, where did the terms come from? Well, early on, people like Dave Washington, Francis Crick, David Washington, those people who are kind of in the forefront of what we now call molecular biology, those people were thinking about this as a language flow, a flow of language from DNA to protein. So the terms then were derived that way. Transcribe means to convert from one form of a language to a different form of the same language. Okay, so if you were trying to write down what I was saying right now, do me transcribing my words. That's where transcription came from. The nucleotide is the same nucleotide language, right? Translate means to convert from one language to another. And so translation came out of the idea that this is a nucleotide language in the RNA molecule, and that gets converted to a amino acid language in the protein molecule. So it's a language flow. And that was the origination of these terms. Right? Now, one thing we have to approach here is what have we learned over the years about this process. Now, DNA, of course, has ATCG, and it has the following, that's what we use as a sequence, right? So, now, the way we write the sequence, convention-wise, is that we put what's called the 5', which is where the phosphate group is. Right, so we have a phosphate group hanging off of here. Okay? We call that the 5' in because that phosphate is on number 5 carbon of the ribose. Over here we have the number 3 carbon of the ribose for carbon 3 primary. Now, so this will be a DNA sequence, alright? And every time you see a DNA sequence, even like online in a database, that's the orientation. It always gets from a 5' to 3' direction. So if we, now, what's the concept of the complementarity? So DNA double-stranded molecule. Therefore, what happens is that the opposite strand is where the base pair relationships come from, okay? And so that when you do that, the 5' end is on this end of the other side, and this is the 3' end. So when the neurons come together, it basically comes together as opposite directions. Textbooks use the term anti-parallel for that. Okay. Now, very important concept here. So, concept 2 would be that all additions to DNA or RNA happens at the 3' OH group, okay? What does that mean? That means that the enzymes which carry out the production of DNA or RNA all have to have that 3' OH group in order to add the next nucleotide. So that becomes, so that's a substrate for the enzyme. Okay, so the enzyme has a substrate that basically allows you to add the next nucleotide going that way. And of course, the process of making DNA is called replication. And in replication, you separate the strands and you make complementary strands to each strand. That's how the replication occurs. Alright, now, so if we are always having to add to the 3' end of a molecule, then which direction is the RNA made? Alright, so the RNA is made, so basically what happens is the RNA has to be made so that the 5' end is here and the 3' end is here, so you can keep adding nucleotides to it. Okay? So, which of these, so we asked the question, which of these two strands serves as a template for the RNA? Okay? And answer that question in the discussion board, and we'll see how many people got that right. Okay. So, another concept in this is that the RNA is a prokaryotic organism, like a bacteria. The RNA and protein are made at the same time. In other words, because of their nucleus to separate the processes, transcription and translation happens at the same time in the bacteria cell. Think about that. That makes sense because bacteria cells have a very short life cycle, and therefore they need to be able to make proteins very rapidly, and therefore they don't really have time to deal with modifications and things like that. Eukaryotic cells, though, have a separate compartment for the transcription process, and therefore they actually can modify the RNA, and therefore have a reason for time for doing different things to the RNA that bacteria can't do. So let's talk about that. And this is, now this is an overview, okay? I did this before, I did the overview, and then we'll delve into it with the PowerPoint, but this is the overview. So if we have a RNA molecule, okay, RNA has uracil, DNA has thymine, right? So different bases in the two, which is very interesting. So your second question to this lecture would be, in the discussion board, what would be the benefit to us from a methodology standpoint? How can we take advantage of the difference in base composition between DNA and RNA? So think about that, research it, and answer that in the discussion question that will appear with this lecture. Now, this is the 5' end, the 3' end by definition, okay? And remember why? It's because we always add to the 3' end, so they always start with the 5'. The first nucleotide has a phosphate group sitting off of it. That's interesting until the eukaryotic cell, because what a eukaryotic cell can do is they can put a 5' guanine group right there and have what we call a cap. Okay? Now, that cap does two things. Well, it does one basic thing. It is involved in transport. If you remove that cap or prevent that cap from forming, then we'll go ahead and inhibit the enzyme that puts that on. The mRNA will never leave the nucleus. So you basically prevent transporters out of the nuclear pore into the cytoplasm. On the 3' end, eukaryotic mRNAs is what we call a burst of polyadenylate, poly-A tail. Now, poly-A tails have been very useful for discovery purposes, because basically they're actually the first gene that was ever isolated, or whatever, the first mRNA molecule that was isolated because they encapsulated in a poly-A tail. A bunch of T's connected to a bead, and they're able to capture the mRNAs by capturing the tail. Nowadays, we want to convert mRNAs to DNA, what we call cDNAs, which stand for copy DNA, or complement DNA. We basically use a poly-T primer to activate the reverse transcriptase reaction, and therefore that's how we make a DNA copy of the RNA molecule. But poly-A tail has some other purposes too. Another purpose of the poly-A tail is to control what we call the lifespan or half-life of the mRNA. Okay, half-life. What does that mean? Well, if you look at and let's say this is time on the x-axis, and this is percent. So this is 100% of the molecules, this is 50% of the molecules, and this is 0% of the molecules. Okay, so basically every molecule has what's called a half-life curve. Alright? A decay curve, if you want to call it that. And so at 50%, that's called the T1/2, or the half-life of the molecule. RNAs are very unstable. They're hard to work with in the lab because there's a special precaution. You have to have tubes and tips that have been treated with RNAs to remove inhibitors, to remove the RNAs that's everywhere. Just with gloves, you have to, you have to be very cautious about it. Almost use almost the amount of a sterile technique approach when you work with RNA. RNA is a pain to work with, actually. So the link to the protease tail is involved with the half-life of the message. So if you put more protease tail, you get a longer half-life, like that. Okay, so this is add, a bunch of age, you get a longer half-life. Now, question 3 for discussion is what would be the benefit to the protein synthesis to have the mRNA last longer, have a longer half-life. Okay, so think about that, look into it, answer that question in the discussion board. What's the benefit for longer half-life? What are you gaining if the longer protease tail give you a longer half-life? What's the benefit to the cell? Alright, so RNA has modifications that we will talk further about, but that basically is the overview of those modifications. Now, let's talk about the layout of the gene for a minute. Okay, so when we talk about a gene, we usually use a straight line to denote the genes, so we don't have to write all the HCCGs out, okay? But basically, in a prokaryote bacteria, we basically have all these have a promoter, okay, and then we have a continuous, what we call reading frame. And that reading frame is what makes the mRNA. It comes out of that reading frame. So the promoter, what does it mean to promote? Means to activate something or to start something. So the promoter is where the enzyme RNA polymerase binds. RNA polymerase binds in the promoter region. So the RNA polymerase binds here and it reads the DNA and makes the mRNA. Now, that's a bacterial gene, continuous reading frame. In contrast, a eukaryotic gene has a promoter, but its promoter is actually more complicated than a prokaryotic promoter. Obviously, no surprise there. But you also have interrupted gene sequences called introns. The introns are large coding sequences that appear to have a role with regulation and also with, things like gene transfer and stuff like that. So what happens is when you transcribe the mRNA, you also, of course, put those intron sequences in there. They go all the way across. And by the way, we call these the exons. Okay? And so we have to splice out the introns. We have to basically remove the introns and put the three exons together. Exon 1, exon 2, exon 3, so E1, E2, E3. We put all the exons together, and now we have a complete RNA molecule that can be translated into a protein. And the other thing we know now, we used to do, so a little bit of a background here. When we were going to sequence the human genome, it was thought that because human cells across the whole organism, if you add all the proteins up across all the different cell types in the human body, you have about 97,000 proteins. 85, 90,000 proteins. Somewhere in that range. When we said, okay, let's sequence the human genome, it was thought we could find 80-some thousand protein coding genes. See, that makes proteins, that makes RNA, that makes proteins. We didn't. We found about 25,000 to 30,000. So you say, what does that mean? If we have, so the question then becomes, the question in the discussion board there becomes, if we have 80,000 proteins, so we have 80,000 proteins and only 25,000 protein coding genes, what does splicing have to do with that? Okay? What does splicing have to do with that process right there? So that would be another discussion question that I want you to answer, and that way we'll have that. Okay, so let me say, so it's 80,000 proteins and 25,000 genes. Okay. 80,000 proteins, 25,000 genes. So the question is, what does that imply? And how can that be that you can make 80,000 proteins from 25,000 or so genes? Okay. That's another discussion question from this lecture. Let me see. Okay. All right. So, the next thing I want us to think about is how this thing is, how we evaluate the expression. Okay? How we evaluate it. So we regulate we regulate these. Now, how do we do that? Well, there's a couple of approaches here, but the one very important approach is that we have proteins that bind the DNA. Okay? We have proteins that bind DNA. These proteins have amino acids would interact with the DNA sequence and allow it to bind to the DNA. That is basically how we regulate gene expression, is by using proteins binding to specific DNA sequences. Alright, example, well, let's take estrogen. Estrogen is a steroid. Okay? It goes across the membrane freely, and it binds to an estrogen receptor protein. Okay? And that goes to the nucleus, and that binds the specific DNA sequence that binds the estrogen receptor. And that binding affects the transcription of the gene or gene that are close by. So we go from, so we bind DNA, by these proteins bind DNA, and they do it specifically. They do it by the protein amino acids recognizing the sequence of the amino acids and of the protein, and the amino acid sequence recognizing the DNA bases and interacting with them, and that's how we, that's the big, on the big picture, that's how we regulate gene expression. Now for a number of years, that was the main focus, was looking at proteins which bind DNA, like the estrogen receptors example, and coming up with schemes to understand what genes are regulated and so on, and how that happens. After we sequenced the human genome, the fact that we had over 25,000 protein coding genes left everybody kind of dumbfounded, to be honest. And there was a project started called the ENCODE project, and the purpose of that project was to see if we could figure out if there were, well, the main focus was to look for these protein DNA interactions across the genome, and the way of identifying where these 25,000 genes were, and also what protein regulated those genes. Well, what they found with the ENCODE project is they found a whole lot more protein DNA binding sites in places where there were not protein coding genes. And that made people wonder, what's going on here? Why would you have a estrogen binding site somewhere in a piece of DNA that is not involved with any of the protein coding genes around? So people start asking questions about whether if we've missed something here all these years. And it turns out we have. We did. We missed a lot of the RNA that actually is being made by the cells. And we had learned about some of that. We knew about things like heteronuclear RNA, which have structural components in the nucleus. We knew about, more recently, we knew about cytosine RNA, which is involved in selecting messenger RNAs in the cytoplasm for degradation. But that didn't explain all these really strange protein DNA binding sites all over the place, even where there wasn't any protein coding genes. And it turns out that what we have now found out is that a lot of the genome is actually transcribed. Different cell types transcribe different amounts. But basically, the bulk of that transcription is not to make proteins. It's to make RNA molecules that regulate the protein coding genes. So we have that whole level now of regulation because now we have RNA molecules involved in regulation of genes. And later on we'll also look at some review articles that will tell us more about that process. So that will be something later on that we'll talk about. But that there really kind of alters the original hypothesis of DNA protein, DNA RNA protein, because apparently it's DNA RNA back to DNA, which didn't go back to protein. So it becomes more complex than we ever imagined this. And so you will see the term that people use now called gene networks. And the gene network is basically all the genes involved with regulating a certain protein coding. And this can be pretty extensive, it turns out, the gene networks. So we'll talk about that a little bit later on. But they can be pretty extensive because they involve not only the proteins that, like the insulin receptor, but they also involve RNA molecules that are involved in regulating the gene. And so also you have the cytosine RNAs, which are involved in helping the teeth with the mRNA gets translated, and so on and so forth. So you have a whole bunch of different levels, and we still don't know much. We still are discovering more about that every day. It's a big ongoing project for us, for cell and molecular biology, to try to figure out what other RNAs are doing. But we'll talk more about that when we get to that point in our thing. The last thing I wanted to do today was to give you a model to understand conceptually, to put this stuff into play. Okay, and my model is this. Now, we know that in the nucleus, DNA is structurally it's structurally attached to histones. We also know that DNA is looped around and it has the scaffold proteins. So we can basically talk about the concept of a neighborhood. Okay, so a neighborhood. And that neighborhood basically would be, let's say that this represents the DNA. Okay, so here's the DNA. I just told you one strand of it. And so let's say that we have, this represents different genes. Okay, so we have different genes going that way. So there's a gene, there's a gene, there's a gene, there's a gene, there's a gene. Okay. We have different genes located in different places in this neighborhood. Now, the idea of, so let's talk a little bit about the nucleus. The nucleus is pretty much a semi-solid environment, which implies that anything that happens in the nucleus is going to happen with minimum effects of diffusion and mostly other types of interactions other than simple diffusion. And we could actually say that in this neighborhood, maybe we only have 2 RNA polymerases. Okay, so we have 2 polymerases, 2 RNA polymerases in that neighborhood. And they're going to go around, almost like training on the track, and they're going to go around and they're going to transcribe these as they come to them. Okay, so here's one, makes a couple on, makes an RNA, goes on down, comes here, makes an RNA, and so on. So you basically are you're reading the RNA, the neurotranscription educational promoters. So basically you have to come to a promoter. So the promoter is what this thing looks for. Okay? It's looking for the promoter. And so when it finds the promoter, it then basically starts transcribing that gene next to that promoter, and then it moves on and moves to the next promoter that's active. And now the other genes may not have active promoters, they may be turned off. So it's just going to deal with that, those promoters that have been activated by the system. If you want to make more of a protein, you have to make more of the messenger RNA within that time frame. And so you have to have a way of recruiting that polymerase. So basically, this guy would go around here, right off the RNA, come back around and do it again, come back around and do it again, and it would make 3 RNA molecules, mRNA molecules. The idea is that the process of raising the gene has to do with recruitment of the polymerase to that promoter over the other promoters which are also active. So there's a point of recruitment going on that is kind of a little model that we can use to understand how these things are put together as far as the ability for the polymerase to know which gene to transcribe and which gene to not transcribe. Because there's only so many polymerases. And so they have to have some way of being selected for that nearby gene to be transcribed. That's where a lot of the protein binding DNA comes in. In other words, say that the estrogen receptor we talked about, that estrogen receptor could be several hundred bases away, or 1,000 bases away, but it's going to, because of the lipid and stuff, it's going to interact with a certain gene promoter, and therefore it's going to recruit or stabilize the presence of different polymerases. And we're going to talk about a paper later on that, a review paper that gives us a model for that. So we'll look at a model for that later on. Okay, this is the overview.